Non-sporing anaerobes. Anaerobes are the organisms that cannot survive even with a little amount of oxygen. So based upon the presence or the absence of the organism, the growth of the organism, we can classify the bacteria into two major types. One is the aerobic organism and the other one is the anaerobic organism. So aerobic organisms are the one that can very well survive in the presence of oxygen, but there are certain group of bacteria that cannot tolerate the oxygen, and the mere presence of oxygen can inhibit the growth of this organism. So these organisms are called as anaerobes, and the importance of this organism was made to us to be known was with the work of Louis Pasteur, who was working with the problem that the French industry was facing with the souring of the wine. So during his study towards the souring of the wine, he found that there are certain bacteria that are able to produce acid in the absence of oxygen, and these organisms were given the name as anaerobic bacteria. And it was Pasteur's organism, which is the Vibrio butyric, was thought to be becoming non-motile when exposed to the oxygen. So the presence of this anaerobic bacteria and the importance of the anaerobic bacteria was made to us known by the Louis Pasteur. It is not only in the fermentation industries or in the atmosphere. These anaerobic organisms are also the ones that are able to cause different types of infection, but they have been often neglected in the laboratory diagnosis. But these organisms are the ones that are mostly related to the anaerobic diseases of the human body, and most of these organisms are pathogenic in nature. It is not because they are uncommon. They are common on the human body, and they outnumber the aerobic bacteria. They are present in every part of the body and have the ability to cause the infection given a chance to them. So the presence of the organism is in 1,000-fold in the intestine when compared to the aerobic bacteria. They are not only present in the anaerobic environment of the human body, but they can also be seen in the mouth and the nasal cavities, and these organisms are thought to be aerotolerant in nature. Now coming to the numbers of this organism, how they usually survive along with the aerobic bacteria, they usually outnumber the aerobic bacteria, and they are present at a range of 10 to the power of 4 to 10 to the power of 5 per ml of the small intestine, and 10 to the power of 8 per ml of the saliva, and 10 to the power of 11 per grams on the colon. So you can see that these organisms are not only present, but they outnumber the aerobic bacteria and have the ability to be pathogenic in nature. Now depending upon the utilization of the oxygen, these organisms can be classified as aerotolerant and strictly anaerobic in nature. So if the organism is able to survive a percentage of 0.3 of oxygen, it is said to be aerotolerant in nature. And most of the aerotolerant organisms are the inhabitants of the mouth and the oral cavity or the nasal cavity, whereas the strict anaerobes are the one that can immediately die even after even a slight exposure to the oxygen. So most of these strictly anaerobic organisms can be seen in the intestine. And these organisms were early classified based upon the colony morphology or the cells or the biochemical reaction and the antibiotic sensitivity testing. Now because of the development of more sophisticated technique, we can classify these organisms into a variety of genuses or the families or the species, and the current classification that is used to classify this organism is completely based upon the molecular typing, that is the DNA based composition and the fatty acid end products, which is the biochemical activity of these organisms. And these organisms are usually classified as anaerobic gram-positive cocci and gram-positive bacilli, and gram-negative cocci and gram-negative bacilli. So coming to the gram-positive cocci, they can be classified as gram-positive cocci and a gram-negative cocci based upon the gram staining. And the organism that are mostly seen in gram-positive cocci are the Streptococcus and the Peptococcus, whereas gram-negative cocci are Bellionella. Next is the bacilli, gram-positive bacilli. Usually they are endospore-forming clostridia and non-sporing, and you can classify the gram-positive bacilli into various types. Most prominent and the most important being the Eubacteria, Propionibacterium, Lactobacillus, Mobilingus, Bifidobacterium, and Actinomyces. Now coming to the gram-negative, these organisms that are included under gram-negative anaerobes are bacterioids, Prevotella. We have Porphyromonas, FUSOBACTERIUM, and Leptotrichia. Apart from this, we also have a spirochetes, which with the example Treponema and Borrelia. We'll be discussing this topic in a separate topic because they are the most prominent organisms that are involved in the sexually transmitted diseases. As of now, we'll be dealing about the anaerobic cocci and anaerobic bacilli. Coming to the anaerobic cocci, these organisms are heterogeneous in nature, usually present in different parts of the human body, and they are gram-positive in nature based upon the gram staining, and they are the normal flora of the mouth, the intestine, and the vagina. Mostly these organisms are involved in the puerperal sepsis and other vaginal infections. And Peptostreptococcus are the organisms that can be differentiated with the Peptococcus because they usually form chains, and they are paired. They can form a short chain or a long chain, and hence they are called as a Peptostreptococcus. They have a structural similarity with the Streptococcus. And Peptococcus are the organisms that are usually the one that forms clusters, and they can be differentiated with the Peptostreptococcus. But recently the DNA based ratio has shown that this Peptococcus can be reclassified as the Peptostreptococcus. These organisms are aerotolerant, that is they are not strict anaerobes, and they can very well grow in a medium containing 10% carbon dioxide. As the normal flora, as discussed, they are usually the one that can be seen in the vagina, mouth, and the intestine, and mostly involved in the puerperal sepsis, which is a childbed fever, and the other genital infections. And they are mostly associated with other type of infections such as the wound infection, gangrenous appendicitis. We have urinary tract infections, osteomyelitis infecting the bones, and abscesses of the brain, lung, or the livers. And this can be very well demonstrated in person suppurative lesions, and we can identify these organisms with the help of gram staining in the pus and the suppurative lesions. And mostly these type of infections are mixed infection, that is they do not cause infection alone, but along with the clostridia or other anaerobic gram-negative organism, they are able to cause the infection, and hence these type of infections are called as the mixed infections. And the most prominent type of species of this group are Peptostreptococcus anaerobiasis, which is the one involved in the puerperal sepsis, and we have Peptostreptococcus magnus involved in the abscesses causing damages to the kidney, liver, and the brain, etc. And other species which are of clinical importance are Peptostreptococcus asaccharolyticus, we have Tetradius, and Prevoti. Now coming to the gram-negative cocci, which is Veillonella, it is a diplococci organism which is a short chain or in group, and it is a normal flora again of the intestine, mouth, or the genitals, and mostly these organisms are non-pathogenic in nature and mostly sensitive towards penicillin. Veillonella parvula can be eliminated immediately on exposure to a small dose of the penicillin, and they are resistant to sulfonamides and the gentamicin. The clinical pathogenic effect is not very well known to be associated with this particular organism. Next is the anaerobic gram-positive bacilli, which are the most important one that can be associated with the different diseases of the human body. And the important one that we'll be studying is Eubacterium. We have FUSOBACTERIUM, Propionibacterium, Mobilingus, and Bifidobacterium. Coming to the Eubacterium, they are strict anaerobes, like we have seen in the Peptostreptococcus, which are aerotolerant. That is, they can tolerate the small amount of oxygen. But here these organisms are strictly anaerobic in nature, and they are the one that are the normal flora of the mouth and the intestine. And the species that are involved in the pathogenic effect of this particular genus is E. brachy. We have Timidium, we have nodatum that are mostly involved in the oral lesions or the dental caries of periodontitis, and E. lentum is the one that is involved in the non-oral lesions. And next important group is a Propionibacterium. Propionibacterium acne is the one that is involved in the causation of acne, which is a skin disease, and this can be demonstrated very well in the blood and the cerebrospinal fluid, and it is a contaminant of the blood and the cerebrospinal fluid. Next is the lactobacilli, which is majorly seen as an inhabitant of the mouth, the nasal cavity, or they are very well associated with the adult vagina, also called as the Dodderlines bacilli. And these organisms are non-pathogenic in nature, except one species, which is Lactobacillus cateniform, which is mostly involved in the bronchopulmonary infections. Next group is a bifidobacterium, which is a pleomorphic in nature, which can change its morphology, and it have true or false branching, and mostly inhabitant of the intestine and the mouth, and is the one that can cause infection in the children who are usually bottle fed. And next is a Mobilincus. These are motile curved gram variable organisms, and the species that are mostly involved in the infections are Mobilincus molaris, Mobilincus curtisi, which is mostly involved in the bacterial vaginosis, along with Gardnerella vaginalis. So it is a mixed infection or a polymicrobial infection that is usually characterized by the thin meladrous exudate with a rotten fish smell. And this rotten fish smell can be accentuated by mixing with KOH solution. And the characteristic feature of this particular disease is the formation of a Q cells. Q cells are usually formed with the epithelial cells with the adherent bacilli. So the presence of this Q cell helps us to know that the disease is associated with the Mobilincus, and it is a polymicrobial disease. Now coming to the anaerobic gram-negative bacilli, the family of that is usually included in this anaerobic gram-negative bacilli is the Bacteroidaceae, with the genuses Bacteroids, Fusobacterium, and Leptotricha. The most common type of infections can be seen because of this Bacteroids, and the Bacteroids are usually non-motile, non-sporing. They are slender rods, and usually they can be grown in a media containing brain-heart infusion, and very well grown in a media with 10% carbon dioxide, and they are usually capsulated, and the presence of the capsule is virulent in nature. Towards this capsule, toxins are usually produced, or the antibodies are produced, which can be easily demonstrated to identify the organism. Now these bacterioids can be classified based upon the saccharolytic activity into asaccharolytic and moderately saccharolytic. So among the asaccharolytic group of organisms, we have a genus Porphyrominus with the species P. gingivalis involved in periodontitis, and P. endodontalis, which is involved in the dental root canal infections. And coming to the moderately saccharolytic species or the organism, here they get inhibited at 20% bile, and the genus involved in this is Privotella, and the species are Melanogenica, and we have Bacillis and Dentacola. Apart from this, we have recent organisms that are called as the bacterioids proper has been identified with the species Bacteroid fragilissus, vulgatus, and Distasonis. Here we have the most important group of organisms that is pathologically like involved, that is a Bacteroid fragilis. And here you can identify the organism in the clinical specimens such as in the wounds, in the suppurative infections, in the CSF, in the blood or urinogenital infections. They can be easily recovered from these areas. And we have a special group of organisms, that is the P. melanogenica. This organism can be identified in the culture by the presence of black or brown colonies. Now the black or the brown colonies is not because of the presence of the melanin, but because of the hematin derivative that is usually seen in the organism, and that gives them this particular or the characteristic colors. And these melanogenica can be isolated in liver abscesses, in the mouth lesion, oral lesions, or in the abscesses associated with the internal organs. And the P. melanogenica can be identified with the help of red fluorescent test with the organism or the wounds or the dressings of the wound that contains this organism. When exposed to the UV light, they give a red fluorescence. That is a characteristic feature of this organism and helps an easy identification of the organism. Next is the FUSOBACTERIUM, another important group of organisms that is involved in the pathological characteristics. They are long, thin and a spindle shape with pointed ends, and they have two species, that is nucleatum, that is mostly involved in the oral infections and also pleuropulmonary infection. If the infection spreads towards the lungs, you can find this organism involved in pleuropulmonary infections. And you have F. necrophorum, which is the one that produces a potent exotoxin that can cause the abscesses of the liver, brain and the other internal organs. Next is leptotrachea, which contains a single species L. bacalis or Leptotricha bacalis, very important organism. It's also called as a Vincent fusiform bacilli or Fusobacterium fusiform. Now this organism has the ability to produce the clinical symptoms which is similar to that of the diphtheria, and hence there is an important thing for us to know that we have to discriminate this organism or distinguish this organism for the treatment of the diphtheria, because this also forms the same type of pseudo membrane which is greyish in color during its infection. So this organism causes a Vincent angina. Apart from that, there is an acute necrotizing lesion that is associated with this particular organism. Now coming to the anaerobic infection. So we have seen about the organism, all organisms that are usually anaerobic and are pathogenic in nature. Now coming to the infections they usually cause in the human body, these organisms, though they are present as a normal inhabitant or the normal flora of the human body, they have the ability to cause the endogenous infection. Endogenous infection is when there is damage or a wound that is caused to a particular area where they usually reside, they enter into that area and then cause the infection. So these infections are usually called as endogenous infections, and but because this organism cannot by themselves initiate infection, they have to have a pre-established lesion in which they can enter and cause the lesion. And there are variety of characters that can help to identify this organism, and this organism can cause infection following precipitating factors. Precipitating factors like trauma, tissue injury, hematoma, or presence of other foreign bodies, or there is a failure in the circulatory system. These are the precipitating factor that initiates the anaerobic infections. Apart from that, diabetes, malnutrition, malignancies are the predisposing factors, or an immunocompromised patient. In those patients also you can see the anaerobic infection being most prominent. And this organism singly cannot initiate an infection. They are usually polymicrobial infection. They are associated with the other group of the organism and they cause the infection. And the presence of this organism in a particular area causing the infection is a localized infection, and if this organism enters into the blood and causes the infection of the other organs, it is called as a bacteremia. So they can cause both localized as well as systemic infection depending upon their presence, whether they are there in the blood or not. And the clinical features that is helpful for us to distinguish between the aerobic infection and the anaerobic infection is that they produce pus which is putrid, pervasive and with a nauseatic odor, except for B. fragilis. So this characteristic smell helps us to identify the gram-negative infections or the anaerobic infections, and moreover they form pronounced cellulitis and there is no toxemia associated with it. That is, they do not produce toxin except for one organism. And the lab diagnosis very helpful for us to identify the anaerobic infection, but because they are normal inhabitant or the normal flora of the human body, cautious interpretation have to be done. The mere presence of the organism in that particular sample cannot help us in identifying the disease, because these organisms are the normal flora, and hence there can be a confusion that can take place during the diagnosis of this disease. disease. So specimen collection has to be done in aseptic condition, and immediately they have to be transported to the anaerobic condition, and minimum exposure to the oxygen have to be seen, because this organism will die on exposure to the oxygen. So the best way to collect the specimen is with the help of aspirations. It is with the aspiration with the needles, and as soon as the sample is collected, immediately it has to be plunged into the rubber cock to prevent the exposure of the organism to the oxygen. And in case of the respiratory infection, sputum cannot be taken as the sample, because the sputum might be contaminated with a different group of organism. So you can go only with the aspiration from the lungs, etc. And once the specimen is collected, you can inoculate onto the anaerobic media, and smear can be done with the help of gram staining. We can identify the gram negative organism or the gram positive anaerobic organisms, and swabs are not advantageous in identifying the organism. And the other method by which we can identify the organism is UV light, that is with the red fluorescence we can identify P. melanogenica, and the most sophisticated one being the gas liquid chromatography. And for inoculating of the organism, we have a special media which have to be incubated under anaerobic conditions. So we have blood agar that have to be incorporated with the neomycin to inhibit the growth of gram positive organisms, and the media should contain yeast extract, hymen, vitamin K, and the incubation should be done anaerobically at 37 degrees centigrade with 10% carbon dioxide. And for fusobacterium, because the organism grows very slowly, you have to incubate for a longer period of time. And simultaneously parallel aerobic cultures have to be made, because most of the anaerobic infection can also be caused by the aerobic bacteria. So simultaneously you have to go with the aerobic culture as well as the anaerobic culture. So this is about the non-sporing anaerobic bacteria that are pathogenic in nature and can cause the infection. Apart from this, we also have the other organisms which are anaerobic in nature and are mostly used in the fermentation industries, such as methanobacterium, etc. But for the pathogenic organism, these are the organisms that are mostly involved in the disease causation.
Completed,